תודה רבה, מר יתום. תוכנית היום תהיה מחולקת לשני מושבים. במושב הראשון ננסה לחסות את החידושים בנושא טיפול גנטי, הן על ידי 4 החברות העוסקות בכך ונמצאות כיום בשלבים שונים של ניסויים קליניים, והן בתחום האקדמי. במושב השני, אחרי הפסקת הצהריים, נתמקד יותר בתרופות המעכבות את התפתחות המחלה ונושאים נוספים. בתום היום נקיים פאנל בהשתתפות המרצים שישמחו לענות על שאלותכם. אורחים ומשתתפים יקרים, לפנינו יום עמוס בתכנים, נודה לכם אם תעמדו בלוחות הזמנים המוקצבים להרצאות ולהפסקות שביניהם. המרצה הראשון שלנו הוא דוקטור ג'ואל שניידר מחברת סוליד ארצות הברית, חברה שהוקמה על ידי אילן גנות, אב לילד חולה דושן, לצורך פיתוח תרופה בריפוי גנטי למחלת הדושן. דוקטור שניידר הינו מנהל טכנולוגיה ראשי בחברה. דוקטור שניידר הוא בעל תואר מאוניברסיטת רוטגרס ותואר מאוניברסיטת ברנדייס. הוא השלים את פוסט הדוקטורט שלו באוניברסיטת הרווארד בתחום תאי הגזע וביולוגיה גנרטיבית. דוקטור שניידר הוא מחברם של מאמרים רבים הקשורים לדושן ולביולוגיית תאי הגזע. דוקטור שניידר, please.
בוקר טוב ותודה שהזמנתם אותי להציג על החברה שלנו סוליד ביוסיינסס, ואני חושב שיהיה הכי טוב אם אני עובר לאנגלית עכשיו, זה יהיה טוב בשבילנו. So good morning, my name is Joel. I'm the Chief Technology Officer at Solid Biosciences, which is a company focused on developing therapies, specifically and only for Duchenne muscular dystrophy. As you just heard from my introduction, Solid was founded by Ilan Ganot, who's our CEO. Ilan is the father of Eitani, who's over there on the left. And while the company was founded because of Eitani and because of the lack of progress on developing therapies for Duchenne and specifically for Eitani's type of mutation, the company is built to develop therapies for, of course, all patients with DMD. And today I'm going to talk to you primarily about our gene therapy efforts. Although it's important to note that during the duration of today's presentations, you're going to hear about how complex the disease is and how while there's excitement around gene therapy and gene editing technologies, there's still a very strong need for complementary therapies that hopefully work synergistically or together with promising gene therapies, even though they're at very early stages. And so because of that, the company is not just focused on gene therapy efforts. We call the development of technologies within our company a 360-degree approach, meaning that we're developing gene therapies, we're developing small molecules, as well as other biologics that will hopefully complement and work together to overall improve the quality of life of patients living with DMD. But today I'm going to focus on our gene therapy efforts. That is our lead program. And importantly, I'll talk a little bit about the manufacturing work that we're doing in order to be able to provide these gene therapies at the large scale necessary to treat the population of patients, which, while rare, is not so rare, as you all know. So as I just mentioned, while our lead program is a gene therapy called SGT-001, we are developing a pipeline of therapies behind that lead program that are technologically diverse, technologically agnostic. So we're excited about gene therapy. There's a long way to go, but we're also developing therapies that are non-gene therapies as well. Small molecules that hopefully address the symptoms that are caused by the absence of the dystrophin protein. We're developing biomarkers to help us understand whether or not any of our therapies are meaningfully impacting the course of disease. And we're even developing assisted devices to hopefully augment and improve on the strength that patients currently have. So as I mentioned before, we consider this a 360-degree approach to solving Duchenne. So because I'm going first today, and because you're going to hear a lot about gene therapy over the duration of the next few talks, I'd like to take a little time up front to really highlight some of the key aspects or the key cornerstones of the development of a gene therapy for DMD, and in general. So gene therapies are comprised of three main components. They're comprised of the gene that you're trying to deliver. In our case, we're trying to deliver a surrogate or a replacement for the absence or mutations within the dystrophin gene. And this gene that we're delivering can hopefully lead to the expression of a surrogate protein that works similarly to dystrophin and retains all of its key functions, but is smaller in size. And that allows it to fit into number 2, which is the viral capsid, or the delivery mechanism that we're using to deliver these genes. We're using a capsid, a naturally occurring capsid, that has shown characteristically a very unique ability to enter into all the tissues that we need to hit with a gene therapy for Duchenne. The skeletal muscle of the appendicular limbs, the cardiac tissue of the heart, as well as the diaphragmatic tissue of the diaphragm, which is critical for respiration. And then finally, we also have to pick our promoter. It's highlighted here as number 3. The promoter drives the expression of the gene that you're trying to deliver. And we're using a promoter that drives high levels of expression in the target tissues that I just mentioned: the skeletal muscles, the heart, and the diaphragm. And so collectively, the gene, the capsid, and the promoter are what we call SGT-001. And in our preclinical efforts, as well as in our ongoing clinical trial, we're delivering SGT-001 systemically or intravenously in a large single dose that accomplishes widespread biodistribution or delivery across the entire body. And we think that it's very critical to deliver these therapies systemically in order to target all of these key tissues. So a little bit more about the 3 characteristics of a gene therapy. The transgene that we're delivering, which I'll talk a little bit more on the next slide, is a gene that is designed to replace the naturally occurring dystrophin gene. I'll talk a little bit about how that was designed in just a moment, but it's important to note that we chose this gene based off of comparative analysis to many different microdystrophin genes. And in our preclinical data, this microdystrophin gene performed uniquely well, and so we chose this microdystrophin to move forward within our construct. As I mentioned before, number 2 here, the promoter, is a promoter that drives expression of the gene specifically in our target tissues, the cardiac tissue, the diaphragm, and the skeletal muscle. And in our analyses, we've seen no expression of the microdystrophin protein in any non-target tissues. For example, the liver, the brain, the lungs, the spleen. We've looked extensively for expression in those non-target tissues and see absolutely no expression, but very high levels of expression in all the tissues that we're trying to hit. And finally, as I mentioned before, we're using a naturally occurring viral capsid to deliver the gene. That capsid is called AAV9, which is one of a number of naturally occurring viral capsids. And AAV9 is unique, similar to other viral capsids that you'll hear about today, because of its ability to enter into the skeletal muscle and the diaphragm and the heart. And it's important to note that all of these classical capsids, AAV9, AAV8, AAV74, they all go across the body. And that's why it's so important to use a tissue-specific promoter, and I'm talking about CK8 now, but you'll hear about others today as well, that drive very high specific expression where you need it to be. So I mentioned that our microdystrophin was chosen based off of comparative analysis to other microdystrophins. And this is a field that has been in development now for over 3 decades. It began with the observations in the clinic of Becker muscular dystrophy patients. Becker muscular dystrophy patients are patients that have a mutation in the DMD gene that leads to expression, so they do have expression, but of a mutated or truncated, shortened form of dystrophin. And there are some patients with Becker muscular dystrophy that have a very mild course of progression. They have a very mild phenotype. And so the researchers that we've been working with for years now have been, well before we even became involved, they've been developing microdystrophins that try and mimic, at the onset, what Becker muscular dystrophy patients express. And then since that initial discovery that those Becker-like genes could lead to improvements in animal models, researchers have been developing novel iterations and novel variants for decades now. And the microdystrophin that we've chosen is called microdystrophin 5. There was a paper published on it by Jeff Chamberlain this past year. In our animal models showed very unique efficacy in its ability to restore function in the animal models, both small and large animals, all models of Duchenne. And uniquely within our construct is a domain that we felt was very critical to incorporate. And that domain is called the NNOS binding domain. You can see it's highlighted in the middle. Yeah, there we go. It's highlighted in the middle here. And the NNOS binding domain is critical for restoring the expression of nitric oxide synthase in the skeletal muscle and the heart. Nitric oxide synthase is a protein, as its name suggests, that is responsible for producing nitric oxide. And nitric oxide leads to, when it's produced during periods of high energetic need, when we're running, when we're exercising, when we're moving very quickly, when we're standing up, nitric oxide promotes vasodilation. So it improves blood flow, it increases the diameter of the vasculature, and leads to proper oxygenation of the muscle beds so that you get normal contractility. Duchenne patients lack dystrophin, as we all know, and because of this lack the nitric oxide binding domain that is important for nitric oxide function. And so patients not only have inflamed and degenerating muscles that are undergoing periods of degeneration and regeneration, they also suffer from a pathology known as functional ischemia. And that's a pathology where the oxygen supply can't meet the demands of the muscle. And so in our animal models, the incorporation of this domain has demonstrated unique functional improvements. So the ability of our preclinical models to maintain high levels of endurance, to resist fatigue. And so we thought that it would be critical to incorporate this domain into our microdystrophin because of these additional key benefits. And then finally, it's important to note that nitric oxide is also a primary mediator of inflammation and fibrosis. And so for all these reasons, we chose to incorporate this into our construct and believe it is somewhat of a unique differentiator in our transgene. And so I highlighted all of the components that went into our SGT-001 construct. As I mentioned, we're using the AAV9 capsid, which is a naturally occurring capsid that is being used in other clinical trials. We incorporated the CK8 muscle-specific promoter that drives very high levels of expression in cardiac and skeletal muscle tissue. And as I mentioned before, that leads to the expression of our specific microdystrophin, which is called microdystrophin 5. And what you're looking at here is immunofluorescence, or it's basically an antibody staining mechanism to demonstrate expression of our microdystrophin after administration. And this is data from one of our mouse studies. All the way on the left side, the leftmost column, is the expression of dystrophin in cardiac tissue and skeletal muscle of a healthy or wild-type animal. And in the next column over, immediately to the right, is a dystrophic mouse, which expresses close to no dystrophin, although they do express some very low levels due to the expression of revertant fibers. And then in the third and the fourth column, I'm highlighting for you expression achieved at the 2 doses that we've entered into the clinic with in our clinical trial called Ignite DMD. The first dose level that we went into the clinic with was 5 times 10 to the 13th vector genomes per kilogram, which we now call our low dose, and that's in the third column. And our second dose, which we announced dose escalation to a few months ago, is approximately 4-fold higher, 2 times 10 to the 14th vector genomes per kilogram. And what you can see is that after systemic administration of SGT-001, at our low dose as well as our high dose, we see very strong expression in both cardiac and skeletal muscle. And so it's these types of data that led to the progression of our program through IND-enabling studies, through both efficacy studies as well as safety studies, to allow us to open up our clinical trial called Ignite DMD, which is being carried out at the University of Florida. But a little more data from the preclinical side before I go into the clinical trial itself. This is more preclinical data, but now from our dog work. We thought it was very important to go into a large animal model of the disease, the dystrophic dog, because it allows us to refine our understanding of how well SGT-001 can work in a large animal model, which is, of course, no true replica or no true mimic of a Duchenne patient, but at least is a large animal and allows us to understand whether or not we can achieve widespread biodistribution and widespread expression of our protein across many different tissue types, but in a large environment, in a large animal environment. And so on the left side of this slide, this is Western blot now. The previous slide was immunofluorescence, and now I'm showing you a more quantitative method to quantify protein expression. But on the left side of the slide, we see examples of expression in two different target tissues. One is the diaphragm, which, as I mentioned before, is critical for respiration. And as we're all probably aware, in DMD patients, the diaphragm is affected at later stages of the disease, leading to cardiopulmonary deficit. But nonetheless, in our dog model, we're able to show at multiple dose levels high levels of microdystrophin expression in the diaphragm, as well as in a skeletal muscle here, one example, the vastus lateralis, at two different dose levels on this slide, 1E14 and 2E14. And as I mentioned before, the doses that we're in the clinic with are 5E13. The 1E14 is approximately 2 times that, and the 2E14 is approximately 4 times our starting dose, and that is the second dose that we entered into the clinic with. And I mentioned before that one of the key components of our construct of SGT-001 is the promoter that we're using, this muscle-specific promoter that drives very high levels of expression where we want it to be. And when we look at non-target tissues, for example here the liver, we see absolutely no expression of the microdystrophin protein in those non-target tissues. And so as I mentioned before, we know that these capsids, these viral vectors, do go everywhere. And that's why it's so important to control the expression of your gene using muscle-specific promoters like CK8 or MHCK7 or others. We did all this preclinical data to really refine our understanding of how SGT-001 could potentially work in the clinic. And there are a number of key different parameters that we explored in small animal models as well as the large animals in both efficacy studies as well as safety studies. And it's covered by 3 different key questions. To the left, how much vector or how much of our virus can we actually get into the tissue? The second question is how much protein is actually produced after we get that virus into the tissue? And then finally, the third most column is how much function or how much efficacy can we observe at those different dose levels? And so starting from the left, what you can see is that as we go up in our dose, we see a very rapid increase in the amount of virus or vector that's detectable in the muscles. And it's important to note that it actually resembles a very non-linear response. So what we may have expected to see is that as we go up in dose, there's a very clear linear increase in the amount of protein we see. So if we increase the dose 2 times, do we see 2-fold more protein? If we go up 4 times in dose, do we see 4 times more protein? And actually, it behaved quite uniquely in the sense that you can appreciate that the slope becomes much more steep as we increase the dose. We call this a nonlinear dose response, meaning that small increases in dose actually had a dramatic impact on how much protein was observed in the muscle. You can see that from starting at doses as low as the 1E13s, which is about 5 to 6 times lower than our starting dose in the clinic, all the way up to doses close to 1E15, we see very rapid increases in the amount of vector detectable in the tissue. And this is a phenomenon that's been talked about for a while now in gene therapy studies. It's described as a threshold effect, meaning that you need to get a certain amount of virus into the muscle dosed before you can achieve really meaningful biodistribution and expression in those target tissues. And this nonlinear dose response is also observed in terms of protein expression in the middle lane, so how much protein is produced. So as we go up in dose, we see that small increases in the dose lead to very large increases in protein expression, or nonlinear increases in the amount of protein observed. And then finally, in the doses, and around the doses that we moved into the clinic with, in our animal models, you can see that we achieve functional benefit above the dystrophic animal models. So in the thirdmost column in black, in the rightmost column in black is our untreated animals, and then going from left to right are increases in dose from 5E13 up to 2E14 and then 4.5E14. And you can see that as we go up in dose, we see a dose-responsive increase in muscle function. And so these are the types of analyses we performed in the preclinical work that we executed in order to understand what sort of efficacy we could achieve before moving into the clinic. And it's important to note that all the preclinical programs and all the pipeline activities that Solid is developing right now go through very similar types of rigorous analyses to really have confidence in our assets before we try and move them towards clinical trials. So now I'll move forward and talk a little bit about the ongoing clinical trial, Ignite DMD. And this trial was initiated last February at the University of Florida under the investigation of Barry Byrne, who's a gene therapy clinician involved in many ongoing gene therapy clinical trials. This is the overall design of that clinical trial in which we're testing SGT-001. We started Ignite DMD with the goal of dosing both ambulant patients as well as non-ambulant patients. And as patients progress in the disease and lose ambulation, they begin to significantly increase in weight. And for those reasons, we chose to begin our trial at a lower dose, balancing efficacy alongside safety. So in ambulant patients as well as non-ambulant patients, would these large amounts of viral vector be tolerated well? And specifically in non-ambulant patients, how would the patients respond to these really massive amounts of viral vector? And I think what's unique, and you'll hear about this a lot today from the other gene therapy programs, is that the doses that we're all in the clinic with are well beyond what is being looked at or studied in, for example, liver-based gene therapy trials. It seems to be that in order to get into the muscle, as I talked about before, you need to go into this higher dose range to really achieve robust efficacy, but also really strong protein expression. But nonetheless, in our starting dose, we chose to take to start at a lower dose to really balance these two points, efficacy alongside the safety. And you can see that alongside every patient enrolled in the trial, in orange, is a matched control patient. In other words, every time we dose a patient, a second patient is enrolled in the trial in what we call a delayed intent to treat cohort, meaning that these patients, after approximately a year of observation, would then be enrolled in the dosing cohort. And what that allows us to do is within our own clinical trial it's not placebo, they know they aren't being dosed, of course, but at least a control cohort that we can follow to really, in a very acute sense, understand efficacy in what we're seeing, hopefully over a shorter period of time. And the primary endpoints for this study are safety, of course, as I mentioned before, and SGT-001 microdystrophin protein expression. So not only are we looking for the achievement of a safe dosing, but also statistically significant improvements or statistically significant evidence of protein expression in the target muscles as well. And alongside those two primary endpoints, we're also exploring a battery of functional assessments, different functional assessments that really elucidate skeletal muscle strength, but also pulmonary assessments, so how well the patients are, of course, breathing, as well as different cardiac assessments, too. And those are mirrored by non-invasive imaging assessments using MRI, both cardiac and skeletal muscle, that allow us to understand how well the heart is working, but in the skeletal muscle, to also understand whether or not we're seeing differences in inflammation or fibrosis or fat deposition. Is there fat accumulation in the muscle too? And so this is the overall design of the trial, and as I mentioned before, we initiated dosing in February of last year. And so in February of last year, as I just mentioned, we dosed our first patients. And our first patient was dosed at the 5e13 level and was observed to have adverse events associated with a decrease in platelet count as well as a decrease in red blood cell levels. Because of those observations, the program was briefly put on clinical hold, and we worked very closely with the FDA to understand exactly what we saw and to remove that clinical hold as quickly as possible. I think it's important to note that since our observations of these decrease in platelets and this adverse event, many other clinical trials in the neuromuscular space have also now disclosed that they see things that are similar as well. And so it's important to note that while these AAV gene therapies show a lot of promise, we still don't completely understand how they work and how safe they are. And that's why it's so important to carry out these clinical trials in a conservative fashion and in a responsible way and you'll hear, of course, from me today, but also from the other groups who are doing really robust and thorough clinical trials to understand not just the efficacy, but also how safe these are and what the tolerability profile looks like. For years, we thought that AAVs were very clean, very non-pathogenic, but we're learning that as we go into these higher doses, while they are clearly tolerated, there's still more to understand about how these function and what it looks like to administer these large doses of vector in patients. So nonetheless, after that adverse event, we moved very quickly back into dosing. And as of a couple months ago, we highlighted that until this point, 6 patients had been enrolled in the Ignite DMD clinical trial. 3 into the active cohort and 3 into the delayed intent to treat cohort. And since then, we also have disclosed preliminary data from our low dose. And that low dose cohort exhibited low levels of protein expression, as highlighted by immunofluorescence, one of the assays I showed you before. And one of the patients showed low levels of protein expression by Western blot. These levels of protein expression were lower than we'd expected. But when we look at our preclinical data, and I'll just go back for just a second to highlight this, you can see that at low doses, and it's in the middle column in the 13 range, there's variability of what types of protein expression you can achieve. And because we saw lower than expected protein expression, we decided to move to a higher dose as quickly as possible. And we announced a few months ago that based on that preliminary data, so low levels of protein expression by immunofluorescence and by Western blot, that we were going to dose escalate to a dose that was fourfold higher than our starting dose, a dose of 2 times 10 to the 14th vector genomes per kilogram. And so in order to do that, we worked closely with the FDA, with the Institutional Review Board at the University of Florida, which of course monitors and regulates all ongoing activities at the University of Florida, and also in consultation with our Data Safety Monitoring Board. The DSMB, or Data Safety Monitoring Board, is a small group of experts in gene therapy and neuromuscular disease that are electively and voluntarily involved in our clinical trial by Solid to monitor ongoing safety and efficacy within our clinical trial. And so before we dose escalate, we